Alo, alo, alo. Okay, testing. Okay, so my mic is okay. Okay, so now is already A3. Okay, so we will start our class now, today. Okay, so for today, we will continue, we will enter the new chapter, chapter 10 lah, which is the fourth chapter of the semester 2. And then yesterday we already finished up the chapter 9 without the for the plant hormone topic, so hopefully you guys can understand it. So for today, we will enter the new chapter, chapter 10, reproduction, development and growth. So actually, in this chapter, we have 4 topics we need to cover: sexual reproduction in human, sexual reproduction in flowering plant, seed germination and also growth. Okay, so firstly, today, we will follow up the sexual reproduction in human. Okay, so I think in reproduction chapter, I think in SPM, the topic that you learn is about the sexual organ, right? Sexual organ, menstrual cycle, spermatogenesis or oogenesis, all that you already learned in SPM. So in STPM, actually is same what you learn in SPM. Totally you copy back that you learn in STPM. So firstly, you will learn about the sexual organ first. You need to know about the human sexual organ lah. So normally, I think this one I don't need to ask. In human, we contain 2 sexes, right? Either you are male or female. So in male, our sexual organ is testis. And then this is the overall structure of male sexual organ. So the main sexual organ that in human is testis. And then we also in male, we also got penis, and also urinary penis, and also got the ejaculatory duct, and then prostate gland, and also bulbourethral gland. So all of these, and also seminal vesicle, all of these are the unique characteristic that found in the human sexual organ. Female, we cannot find it. For like testis, penis, and prostate gland, bulbourethral gland, and also seminal vesicle, all these structure you cannot find in female one. This is the spatial structure that only can be found in human only. Okay, next is about female sexual organ. So in female sexual organ, the main organ is ovary, right? The main part structure is ovary. This is the place to produce ovum, to produce the egg lah. I think in general terms we call egg, but in biological term we cannot say egg. You need to say is the produce ovum lah. So over, and also got oviduct, or another name in others book they say fallopian tube. So both are acceptable. You can use oviduct or fallopian tube are acceptable. And then you also containing uterine wall, and uterus. And then we also contain vagina. So vagina is the place that the penis to come in during the sexual intercourse action lah. Okay, so this is the overall structure about the sexual organ. So I think this one everyone should very familiar with it lah. Okay, wait Okay, next we will go to the another topic is passage of sperm. Okay, so this is the in our syllabus. Before you learn this, you need to know the overall structure of sexual organ lah. So this is in our the first outcome that by MPM about the passage of sperm from the male until female. So this is the how the sperm can be swim, you can say, can be swim from the male sexual organ into the female sexual organ during sexual intercourse activity. Okay, so this is the overall general process. So I think a general process. Okay, so normally we know that sperm is produced in the seminiferous tubule through the spermatogenesis, right? So later we will learn about the spermatogenesis process, but for now you should know that sperms actually is produced within testes with a structure we know as seminiferous tubule. So you see this diagram, so this is the testis, right? Testis. So within the testes, there are many tubule we call as seminiferous tubule. So this is the place where the sperm are produced. Okay. So once the sperm are produced, they will pass into the chorioduct structure we call as epididymis. So this is the place for the sperm to mature about 3 weeks. So normally generally is about 3 weeks. So this is a place about the maturation of sperm Okay. After that, when there are the ejaculation process, so normally this is a male sexual activity. So if you do a jerking, or another terms in general term you call as jerking lah. So in ejaculation process or jerking process, the sperm will transport through the vas deferens. So you see this structure. So once the sperm when the male does not do a jerking process, or we call as ejaculation, if no ejaculation process occur, the sperm will store in the epidermis first. So once there are the jerking process, the sperm will pass through the vas deferens tubule. Okay, so when they are passed through the vas deferens, the vas deferens will extend until the behind of the urinary bladder. So it is like a tube lah. We can say that vas deferens is like a tube until it reach the urinary bladder. Okay, so you can see that there is like a tube. Vas deferens is like a tube that it will pass through until it reach the urinary bladder at here. Okay, so after that they will have the another structure we call as seminal vesicle, and then the ejaculatory gland, and also prostate gland, and also the bulbourethral gland. So all of these have their own function. So what are their function? So normally if you see this, there is a joint of duct from seminal vesicle that will form the ejaculatory duct. So normally this duct and the seminal vesicle will secrete the semen, which is the fluid that will be ejaculated, and then also the fluid that responsible for sperm to swim. Okay, and then normally the main function of seminal vesicle is to secrete the semen lah, and also normally secrete the semen. So normally the semen is yellow color, and then alkaline, and then contain mucus, fructose, and also acidic compound. And then also the prostate gland also can secrete semen and directly into the urethra, which is milk and thin. So both of them are secrete semen, but the component are different lah. Okay. And then another gland also will secrete clear mucus to neutralize any acidic urine remaining in urethra. So if you know that the passage of the sperm is similar to passage of the urine in the male structure. Okay, so normally this bulbourethral gland is to secrete mucus to neutralize any acidic urine remaining in urethra. So E1 is to prevent the sperm be killed by the acidic component that found in the urethra lah. Okay. After that, once the semen is already secreted, sperm was starting to enter. Will use this fluid semen, this fluid to swim, and then transported into the urethra through the ejaculatory duct, and then to the penis. So penis is our excretor copulatory organ that deliver the sperm into the female reproductive tract, which is the vagina lah, and then vagina. Okay, so normally normally during the sexual intercourse activity in male, so normally your penis will fill in the blood, so your blood pressure at the penis is very high. So that is why that during sexual intercourse activity, the male penis will be in very red color compared to that does not do sexual intercourse activity. So during sexual intercourse activity, your penis will be have a red color. Why? Because all the blood will be rush into the surrounding of the penis. So surrounding your penis will containing the blood, containing high amount of blood lah. Okay, and then the pressure of the blood also very high. Okay, so therefore your penis erection occur when penis is increased in length, diameter, and firmness to cause the penis can be inserted to vagina. So during the investing state, or we can say investing state, or you are not doing the sexual intercourse activity, your penis structure is like this. But when you are having a sexual intercourse activity, your penis erection will be occur, meaning that your penis will become straight and become more higher lah, compared to that does not have the sexual intercourse activity. Okay, after that, lastly, during sexual intercourse activity, sperm will swim within the semen toward the cervix and then toward the oviduct for fertilization. So when they are at the same time in the oviduct or fallopian tube containing the ovum or secondary oocyte, then fertilization may occur. But if no secondary oocyte in the oviduct, so no fertilization occur. So this is just a normal sexual intercourse activity only. So that's all for the passage of sperm in female lah, from female to female organ. Okay, after that we will go through the spermatogenesis. This is the process to produce sperm in the seminiferous tubule. So as mentioned before, we know that sperm is produced within the testes, and then within the testes there are many tubules we call as seminiferous tubule. So this is the place where the sperm is produced. Okay, so I think the spermatogenesis process, everyone should be very familiar with it lah, because it's totally same in that you learn in SPM. So normally in spermatogenesis process, the origin compound in the seminiferous tubule, the cell are we call as primordial germ cell. So this is the germ cell that containing inside the seminiferous tubule for the origin cell. So this origin cell will undergo mitosis process to form the spermatogonial stem cell. So this cell actually is within the embryo. So after that, they will produce a stem cell we call as spermatogonial stem cell. So this is the stem cell. Stem cell meaning that this cell can be replicated more and more, can be replicated forever. Once you are surviving, these cells will be replicated as well. So that is why that one of the reason that the sperm, even though you are very elderly, like 70 years old, 80 years old, if you are a very healthy person, you are the male still is able to produce the sperm because of this stem cell. Why? So you see this kind of error. So this stem cell can be replicated more and more until along the lifespan of the male lah. Okay, so this stem cell, they will undergo mitosis process to form spermatogonium. So this spermatogonium also will undergo cell division, mitosis process to form primary spermatocyte. Okay, after begin the primary spermatocyte, meiosis will occur. So we know that you already learned in SPM, meiosis will occur twice, right? There are meiosis 1 and meiosis 2, and then primary spermatocyte will undergo meiosis 1 to form secondary spermatocyte. So for begins secondary spermatocyte, your chromosome number is already N. So meaning that in human we got 46 chromosome, right? So now secondary spermatocyte, it containing only 23 chromosome. So after secondary spermatocyte, they will undergo meiosis II also to form spermatic. Okay, so this spermatic actually is not the sperm cell yet. It will undergo the cell differentiation process to form the major sperm cell. And then after that, it will release to the lumen or seminiferous tubule, and then go toward the epidermis for maturation process. So this is the overall process of the spermatogenesis, so more easily lah. And then okay, this one is the hormone regulation about the spermatogenesis that I take from the Campbell. But I think this one is not very important but as for information. So actually male, we know that the male hormone, the main hormone is testosterone. Testosterone is the key hormone key male hormone to contribute the spermatogenesis one. But in male, they are actually containing FSH and also LH, follicle stimulating hormone and luteinizing hormone. This one I think you only found in female, right, in SPM. But actually female also able to produce these two hormones, but the amount are very little compared to female. Because these two hormones are responsible to secrete testosterone. LH is responsible to secrete testosterone, and the FSH is responsible to secrete inhibin, and also inhibin 1. So normally this is a feedback to tell you that which time the cells will undergo the spermatogenesis to produce sperm, and which time will not. So there are the negative feedback mechanism lah. You will see this negative feedback mechanism, meaning that it will start to inhibit the anterior pituitary gland to secrete these two hormones. Okay, and also it may also to inhibit hypothalamus to secrete GnRH2. Okay, but this one is not very important. The most important is this. This one is mostly important. Okay, and then this is the explanation or the explanation about the spermatogenesis. Okay, so this is the sperm structure. So normally sperm structure containing three main structure about the head, and they got the middle space which of the mitochondria, and also tail structure. So normally tail it's play the role to swim or another name we call as flagellum. So flagellum actually is play the role for the swimming process. And then the head is play the role in fertilization process, because it containing the nucleus membrane sac called acrosome. This is the acrosome, which is found in the sperm head and play the role in the fertilization process. Later we will learn it about the mechanism of fertilization process. And then in the middle we containing the mitochondria too. So this mitochondria actually is provide the energy to the tail, or we call as flagellum, for the swimming process. Provide the movement of the sperm, so that the sperm can swim from the vagina, from the female vagina, until it reach the female fallopian tube. Okay, next we will talk about the oogenesis, which is the formation of the ovum in the female. So compared to spermatogenesis, it will be more complicated in female. So female is more complicated compared to male one in terms of oogenesis, or formation of the ovum and sperm. So oogenesis, as we know that initially it also initial from primordial germ cell too in the embryo. But the differences is, in sperm it will undergo cell division to produce spermato stem cell, right? Spermatogonial stem cell. Now in sperm, spermatogonial stem cell, this is stem cell, so it can replicate along the lifespan one. But for female, it will not produce stem cell. It will produce the structure we call as oogonym. So it is not as the sperm cell. So meaning that if there are how many germ cell in the embryo, so there will how many oogonium will be produced. So it is not a stem cell, meaning that oogonium cannot replicate more and more along the lifespan. So that is why the female got menopause. During menopause, there are no menstruation process anymore, meaning that the female body does not containing any ovum already. Okay, so this is the first step. Okay, so once the first step oogonium is produced, it will undergo the mitosis process again to form the primary oocyte. Okay, so ovum is located within the follicle. It's not like a sperm. Like a sperm does not require follicle, but for female they require follicle. So normally this egg is within the follicle one. Okay, so normally in oogonium it will undergo mitosis process to form primary oocyte, and then the follicle will be grow until become the growing follicle lah. Okay, so inducing the primary oocyte, the interesting is it will undergo meiosis 1, but the meiosis 1 process will be stopped at phase 1. And then this process, all of these is happening in embryo state only. So meaning that these cells in female compared to male, it will only form the spermatogenesis is continue along the lifespan from the spermatogonial stem cell until it reach the secondary oocyte. However, in female, the oogonium and the primary oocyte formation is occur in embryo. So all of the during the pregnancy, if you are in pregnancy, all the female sexual ovum will be produced until the primary oocyte, then stop. It will stop at the phase 1, then it will stop until it will continue again when you are reach the puberty age. When the female reach the puberty age, so normally is 12 years old or 12 years old or 13 years old, like that lah. So when they are reach the puberty age, then meiosis 1 will be continue from phase 1. So it will complete the meiosis 1 and then go to the meiosis 2, and then it will form the secondary oocyte, and then this secondary oocyte, meiosis 2 is not completed yet, it will stop at the metaphase 2. So once it enter the cells and complete the meiosis 1, and then it will enter phase 2, and then go to the metaphase 2, and then it will stop. So once it stop, it will form the structure we call as secondary oocyte. But only one ovum can form secondary oocyte, and another one is we call as first polar body. This one will be discarded in the body. This one is not a egg lah. So that is why that in female there are only one ovum only, or one ovum at once. Karena dia tak akan not like the male, it can produce two secondary oocyte here, so only one only. So after that, the secondary oocyte later it will wait either got fertilization or not. So if no fertilization process, this ovum secondary oocyte will be released out through the menstruation. But if there are fertilization, meiosis 2 will be continue, and then complete the meiosis 2, and then to form the fertilized egg. So this is more spatial compared to male. Okay, so this is overall process of oogenesis. So any questions so far? Okay, so For now, wait a moment. I need to settle something first. 哎呀，阿雷，做咩有蝴蝶跑进来的？怎样抓？开窗，快点跑出去！去！喂，出去啦！孩子晚上不能开窗。我不想杀死你，快点跑！自己醒某一点，自己出去啦！哎，出去啦！我老喂，还是要开门。来，出去那边。是，跑去那里。出去！我想杀你，我不想被人丢，出去啊！Okay, so this is all about the process of secondary or formation of the secondary oocyte lah. No, oogenesis. Okay, so this is very highly related. Later we will talk about the menstruation. So it's highly related. So as I mentioned before, secondary oocyte it will produce will produce when the female is reach the puberty age. So they will complete the meiosis 1 and then enter meiosis 2 and then stop at the metaphase 2. And then it will only complete the meiosis 2 when they are fertilization process. So during this time, actually ovulation is occur already. The secondary oocyte will be released through the ovulation. So meaning secondary oocyte is now released from the ovary already. So you see, it will refer to back to the organ structure, female organ structure. It will release from the ovary and then go to the fallopian tube. Okay, when they are fertilization, meaning when they are sperm enter, then they will conduct a fertilization and then it will complete the meiosis 2 to form fertilized egg. If not, the secondary oocyte will be released out from the female body through the menstruation process, and then at the same time your follicle is still developed, and then once the secondary oocyte is already released from the ovary through the ovulation, the follicle will form the corpus luteum. Okay, so this corpus luteum later it will have a function that play major role function in the pregnancy. So if there are no pregnancy process, meaning there are no fertilization if there are no fertilization process, then the corpus luteum will be degenerated and then to form back again the primary follicle again. So this is the overall process of oogenesis. So oogenesis will be occur once in a month. So that is why menstruation got the female will undergo menstruation once in a month. So next we will go to the menstrual cycle. Okay, so this topic I think you also already learned in STPM Bio. So there are normally in menstruation you are very famous in these 5 hormones. These 5 hormones are very important during the menstruation, especially these 4: follicle stimulating hormone (FSH) and luteinizing hormone (LH), progesterone, estrogen, and also gonadotropin releasing hormone. Okay, so FSH and LH are secreted from anterior pituitary gland, and progesterone and estrogen are secreted from corpus luteum, and then this gonadotropin releasing hormone is secreted from hypothalamus. So the site of the secretion you also need to know. Maybe in objective question they will ask you. Okay, so this is the overall menstruation process. So normally in first stage of the menstruation process, hypothalamus was start to secrete GnRH, meaning is gonadotropin releasing hormone. It will secrete the GnRH RH first, and then this GnRH will stimulate the anterior pituitary gland to secrete the FSH and also LH. But for now, it will secrete more FSH compared to LH. But actually, at the same time, they both will be secret, but FSH will be higher amount compared to LH because it need to follicle stimulating hormone, meaning that it need to stimulate the growth of the follicle first. It need to stimulate the growth of the follicle, and then now your ovarian cycle now is the follicular phase. So the naming of the cycle is we call as follicular phase, and then the naming of the menstruation cycles is we call as menstrual flow phase. So actually, this is the time where the menstruation is formed. Meaning that this is a very difficulty time period for female because this is a breeding time. But at the same time, your hypothalamus will start to secrete GnRH to prepare the new cycle of the menstruation. So since the concentration of estrogen is low, this inhibit the production of FSH and LH. So although GnRH will start to secrete the anterior pituitary gland to secrete the FSH and LH, but since your estrogen now is very low, your estrogen, another name you see this they call as estradiol. So this is American style American terms. But in British term we use estrogen. So yeah, so estradiol is equal to estrogen Okay. So due to the estrogen now is very low, so your production of FSH and LH will be inhibited. So it will not secrete in higher amount. So you see this diagram, FSH and LH will not secrete in higher amount. So when the follicle secrete more estrogen, cause the estrogen level to increase. So the estrogen will stimulate the GnRH, causing more GnRH is released. Since although FSH is inhibited, but at the initial state, since there are FSH is starting to secreted now, so FSH will start to develop the follicle of the growth, and then the development of follicle was starting to secrete estrogen. It will secrete the little amount of estrogen first, little amount of estrogen. So that is why that you see the estrogen level is high at here, and then this one will cause the FSH and LH starting to increase. Why? Because estrogen will stimulate hypothalamus to release more GnRH. And then this GnRH will stimulate anterior pituitary gland to stimulate high amount of FSH and LH, especially LH. So normally higher amount of LH will secrete will stimulate the ovulation process to release the secondary oocyte. So the secondary oocyte will release from the follicle now. So the high level of the estrogen will also increase the sensitivity of GnRH to LH, thus the level of LH also increase. So not only FSH but LH will increase more higher compared to FSH. Okay, so when the follicle become major, it will enlarge to form a bud. It will more bengkak at the surface of the ovary. So you see this diagram it will more bigger, more larger And then the high level of LH will stimulate ovulation, which the secondary oocyte will be released from the Garfian follicle. So actually it will form the Garfian follicle and release out. And then ovulation cause the follicular phase, and now it will enter the luteal phase. And then at the same time, your uterine cycle we call as proliferative phase. Okay, next we go to here. So if no implantation, go to here. So now when the Garfian follicle is released out the secondary oocyte, so LH hormone will start stimulate the ovulation, and also it will stimulate the rupture follicle into the new structure we call as corpus luteum, that just now I said. And then this corpus luteum play the major role to secrete estrogen and also progesterone, especially progesterone. So to inhibit the secretion of GnRH. So the corpus luteum will secrete the progesterone and estrogen, which will inhibit the secretion of GnRH, so that to prevent the another cycle. So it need to prevent the another development of follicle and also the ovulation process. So it will inhibit the higher amount of progesterone and estrogen will inhibit the secretion of GnRH. So since no GnRH, so anterior pituitary gland will not stimulated, so no FSH and LH lah. Okay, LH lah. So since the secretion is inhibited, so FSH and LH are also inhibited. So it prevent the maturation of another egg. So your another egg in terms of secondary, it will remaining on the primary oocyte. Okay, if there is no pregnancy, the low level of FSH and LH will cause the corpus luteum to degenerate and decreasing level of progesterone and estrogen. So this one, if there are no fertilization process, corpus luteum will be degenerated, and then degenerated. Once the corpus luteum is degenerated, your progesterone and estrogen level will be declined also. So you will see like this. So it will enter another cycle again, because corpus luteum play the major role in secretion of progesterone and estrogen, right? So now corpus luteum is disappear. So progesterone and estrogen cannot maintain the high level lah. So it will down, it will decrease the level, and then increase the level, and then decrease, and resulting the menstruation process, which is a breakdown of endometrium wall. Why? Because we know that estrogen and progesterone play the major role in development of endometrium wall. So you see this endometrium wall. If you found that when the estrogen is increased, your endometrium will start to increase already. And then after that, when you reach the progesterone wall, progesterone level is in the peak, your endometrium wall is the thickest now. Okay, so the phase now in menstruation cycle now is ovulatory phase. For ovulatory phase, when the estrogen level is increasing before the ovulation process. Okay, so the phase now, the mean that follicular phase is coordinate to the proliferative phase, and then the high level of progesterone and estrogen that's secreted by corpus luteum would stimulate the maintenance and further development of endometrium wall. So estrogen main function is to repair and growth of endometrium wall, but progesterone main function is to maintain and further develop a endometrium wall. So actually both lah, but progesterone play the major function. Okay, this will also enlarge the uterus and the growth of the endometrial gland. So actually you see this there are reach of the blood vessel there lah. Okay. So now when the progesterone is in the peak, and then to increase the thickness of the endometrium wall, now it will go to the secretory phase in the uterine lah. And then this means that luteal phase of the ovarian cycle coordinate the secondary phase of menstrual cycle. Okay, so if there are two situations, either got fertilization or no fertilization. So if no fertilization, I already mentioned, corpus luteum will be degenerated, and then cause the decline level of the progesterone and estrogen, and will cause the artery in the endometrium wall to constrict. So it will break down the endometrium wall, because progesterone and estrogen main function is to maintain the thickness of endometrium wall. So now progesterone and estrogen is already disappear, already declined. So your endometrium wall cannot maintain its thickness, so it will start to break down. So firstly, it will break down through the uterus blood vessel, starting to constrict, and then the endometrium wall will largely disintegrate by releasing the blood along the endometrium wall, and then the menstruation will occur. So it will reach the menstrual flow phase. So the menstrual flow phase will stimulate the new set of the ovarian cycle to begin to grow. So this it will repeat and repeat again in a period of 28 days. So that is why that we say that the female will face the menstruation once a month. So because the period is about 28 lah, but need to see different individual. Different individual will have different periods. Some will have until 31, 32. Some maybe more earlier, 27, 26 like that. But cannot too earlier and cannot too long lah. If cannot too earlier and too long, maybe got something problem in your hormone level lah. Okay. So this is all the process involved in the menstrual cycle. So the most important is at the end of the phase of the menstruation. Either got fertilization or no fertilization. If got fertilization, your progesterone and estrogen will be maintaining at high level. It will not drop down. And then no menstruation as well. But if no fertilization, progesterone and estrogen level will be decline, and then resulting in the menstruation lah, and then it will continue back another development of follicle and also development of and completion of the oogenesis process. Okay, so that's all for the about the menstrual cycle. Okay, so this is overall lah, another books process. So actually similar what I say before, you can read yourself. Okay, now we go to the human embryo development. Okay, just now we already mentioned about the sexual organ. It is a totally continuous process, and then if you want to have the human, you want to get the give a birth, you need to have a sexual intercourse activity first, right? Male and female need to do a sexual intercourse activity, and then the sperm is already flow into the fallopian tube and undergo fertilization. Then you can proceed to human embryo development. If not, then no human embryo development as well lah. Okay, so this is the first process. First process, you need to have a fertilization first. But before fertilization, of course, you need to do a sexual intercourse activity first. If you not do that, then no fertilization. But now you can use the manual fertilization, we call it in vitro fertilization, without doing the sexual intercourse activity. But in general way, we will do the sexual intercourse activity resulting in fertilization process. So once the fertilization process is completed, it will contribute to implantation process. So you see this. So normally the morula and blastocyte, all these, you see this, it implant. This one is we call as implantation process. After that, after implant, it will go through the two process we call as gastrulation and oogenesis during pregnancy. So after fertilization, this one process is actually we call as each female is actually already pregnant lah. This is the early stage of the pregnant. We call as first trimester, mister, and then this second mister, and then this third trimester. Okay, so this is early stage, it is secondary stage, it is early thirdly stage, which the organ is formed, and then finally is parturition. This is the process of the birth, the birthing process from female. Ini kelahiran anak lah, kelahiran anak daripada ibu dia. So all of this is the new things that added in the STPM syllabus that you never learn in STPM. I think fertilization, everyone know what is the terms meaning, but in SPA you not learn about the mechanism. Okay, so you will learn all this mechanism one by one lah. Okay, so this is a summary process or implantation. So I already mentioned that when there are the sexual intercourse activity at there, sperm was starting to swim at here from the vagina until it reach the cervix, and then you go to the uterus, and then it will go reach to the fallopian tube. And then at the same time, if you want to have a fertilization process occur at here, meaning that ovary will start to release the secondary oocyte. Ovulation occurs, release a secondary oocyte. Enter the fallopian tube, and then at here, if there are the sperm at the fallopian tube, then fertilization will be occur, maybe occur. If not, then no fertilization, and then this secondary oocyte will be go until it reach the uterus, and then it will attach to the implant. It will either go to the uterine wall. If no fertilization occur at here, then the secondary oocyte will directly release out from the female sexual organ and then resulting in menstruation process. But if there are the fertilization process occur now, so there will fertilization occur. So fertilization occur, and then it will go to the cleavage process. So now your cell will be undergo cleavage process. It will become 2 cells, 4 cells, 6 cells, 8 cells, 16 cells, 32 cells, until it becomes 100 cells, until it go through the forming a blastocyst. Forming the whole structure we call as blastocyst. And then this blastocyst, when it reach the uterine wall, it will implant into the endometrium wall. So this is the process we call as implantation process. So this is the overall process. Okay, now we need to learn about the fertilization process. Okay, so this is the overall general law of the fertilization process. Okay, so as I mentioned before, we know that there are the sperm containing the key structure we call as a chromosome. So a chromosome actually is the key structure to provide let the sperm can be penetrate into the ovum. So this actually is the fertilization process in the jelly inside the jelly. In the jelly, it is not in human, but almost similar. Later I will explain about the inhuman one. So okay, in human, normally the fertilization process begin with the capacitation first. So this is a capacitation, meaning that the sperm undergo capacitation. It is a maturation process and secretion process of the sperm in female reproductive tract, and then this sperm will more motile and able to enable the sperm to undergo fertilization. So after the capacitation, the sperm will move through the corona radiata using the hyaluronidase enzyme. So this one we call as in human ovum compared to this jelly. This one is jellyfish. In jellyfish they are actually containing jelly called. But for human, we does not containing jelly. We will containing the cell we known as the corona radiata. So there are many cells actually founded at the surrounding of the secondary oocyte one. So the sperm need to pass through all these cell first before it reach the secondary oocyte. So it will release the enzyme we call as hyaluronidase enzyme. Okay, enzyme. So after they already using the enzyme degrade all these cells, they will enter the sperm. So the sperm will bind to the ZP3 molecule, which a receptor in the zona pellucida, which trigger the acrosomal reaction. So it will trigger the acrosomal reaction by binding the receptor on the zona pellucida. Here, so this receptor we call as the P3 the P3 receptor. Okay, so this one acrosomal reaction is triggered by the acrosome. So this acrosome will release the hydrolytic enzyme to digest the zona pellucida. So zona pellucida is the outer surface membrane structure of the secondary oocyte to make sure that the sperm can penetrate into the secondary oocyte to release the nucleus . Okay, so the sperm head with nucleus will penetrate to the cytoplasm of the egg. So you see this diagram. So similarly, firstly it need to contact first contact first, but in the another type of animal, it need to digest the jelly cord first lah. But in human, we need to digest the gena pellucida first. But after that, it will go through the echosomal reaction. So similar to human, it will release the enzyme first, and then enzyme to make a hole in the jelly cord. So your going actin filament form the echosomal process, which protrude from the sperm head and throw through the jelly cord. So protein on the surface on the ecosome bind to specific partner protein in the egg of the plasma membrane. So the general function is to ecosome is to release enzyme to hydrolyze out the surface membrane of the secondary oocyte to make sure that the nucleus can be passed through into it. Okay, so once they already release out of the enzyme, when the sperm head is already penetrate the cytoplasm of the egg, sodium ion channel in the egg plasma membrane was starting to open. So this one will cause depolarization process. Yeah, so this is actually is an action potential process. Action potential process that you learn in chapter 9, right? You still remember about the action potential process. So why we want to add the action potential? Because it need to prevent additional sperm diffuse again to the egg. So this one is act as the fast block of the polyspermide. So this one is one of the mechanism that in the human to prevent their second sperm penetrate into the ovum. You will still remember that in the rules of the fertilization, only one sperm can be penetrate into one secondary oocyte only. There are no 2 sperm, there are no 3 sperm, there are no 4 sperm can penetrate to the secondary oocyte at the same time. If they happening like that, so normally there are something problem inside the female body lah. There are something problem at there already. So if general condition, they will not have a problem like that. Okay, so at the same time also, the binding of the sperm to activate the signal transduction pathway, which trigger endoplasmic reticulum release more calcium ion into the cytosol. So if you still remember about the chapter 9, I already explained what is the meaning of signal transduction pathway. So when the binding of sperm, it already binding, right? I already mentioned it bind to ZP3 molecule. So it will trigger signal transduction pathway to secrete the enzyme, and then it will trigger the series of the action But the series of the action you don't need to know. You just want to know that it will trigger the action of the signal transduction pathway. The finally response is it will trigger the endoplasmic reticulum to release more calcium ion into the cytoplasm of the secondary oocyte. So increased level of calcium ion will trigger the cortical granule within the egg fused with the plasma membrane of the egg. So you see that there are many cortical granule at the cytoplasm of the egg one. So once the already trigger the signal transduction process, the calcium ion is passed through into the cytosol of the secondary oocyte. Secondary oocyte, it will trigger the cortical granule within the egg fused with the plasma membrane. So this cortical was starting to fuse to the plasma membrane of the secondary oocyte. Why the cortical granule want to fuse on the plasma membrane of the site? Because this one cortical granule was secret its content. It will containing the contain normally is enzyme to break down to create the space between the plasma membrane and zona pellucida layer like this. Dia nak macam cut down lah, macam buat satu longkang macam tu, macam satu gap layer like that. Want to create a gap layer like that. Why you want to create layer like that? similar as the function I say before, you need to prevent another type of sperm, another sperm penetrate to the secondary oocyte again. We don't want like that. You only want to ensure that only one sperm penetrate one secondary oocyte. So this one is act as the slow block to polyspermide. Slow block to polyspermide. Block to polyspermide meaning it need to block to the polyspermide, meaning many of sperm penetrate into the secondary oocyte. But this one is more slowly lah compared to this one. This one the depolarization process is fast block. Because it trigger immediately. When once the sperm is penetrate into it, it will trigger immediately. But for this one, it need to take a moment first. You need to trigger signal transfer process first, and then release calcium ion first, and then baru cortical granule was fused to the plasma membrane of the secondary oocyte. So this is more slowly lah compared to the depolarization process. Okay, so once the already create the gap, create the space between the plasma membrane, so it will trigger the cortical variation. So cortical variation make the zona pellucida away from the egg and harden the zona pellucida. So now your fertilization envelope is formed. So now your sperm nucleus is totally entered into the into the cytoplasm of secondary oocyte, and then the fertilization process is completed. So this is all of the fertilization process. Okay, so as I mentioned before, if you already completed the fertilization process, next you will have implantation process. Okay, so you recall back this. I already say fertilization process is completed here. And then after that, you will go through the fertilized egg. Was starting to move through the fallopian tube until it reach the uterine wall, and then it will go through implantation process. So all of the process you call as implantation lah. So you will see one by one. So normally in implantation, it will including the cleavage stage. So normally when the egg is fertilized, it's like this lah. In microscope, you will saw like this or like this lah. This one is called color one, more color. This one is veil structure like this, like that. So after that, it will undergo cleavage state. The fertilized egg was starting to divide many cells. Now it will go to 4 cells, and then egg cell actually got 4 cells, egg cell 16 cells, 32 cells, 64 cells. It will doubling the cell stage until it reach the bastula, or in another name we all as blastocyst lah. Blastocyst. It will form the blastocyst. So this is the very the final cleavage product in the cleavage stage, blastocyst. So this term you need to memang kena betul-betul tahu. So yeah, secondary oocyte, and then normally during cleavage both G1 and G2 is skip. So you still remember about the mitotic cell division process. We got G1, G2 and mitosis, and then got and also S phase, right? G1 S phase, G2 G1 phase, S phase, G2 phase, and also mitosis phase. You still remember about the cell division process that you already learned in SPM. Okay, so normally during cleavage, because it is a rapidly process, so G1, G2 are normally skipped lah. So it will directly go to mitosis process. So that is why that you see this, the size of the cell are not increased. Actually, the size are same. Just that inside the one cell, here there are many cells here, but the size are same. Okay. So the cleavage causes the cytoplasm of the fertilized egg divided into many smaller cells called blastomere. And then after 3 days to 4 days after fertilization, it will call the morula. So normally molar, but the final product is recalled as the blastomere lah. So actually bastula. The bastula is formed that consists about the blastomere. So I mean bastula at here, blastocyst. Okay. So this is the final product like this. This is the final product, or you see this. So this is recalled as blastocyst. And then inside the blastocyst, there are many structure first. Many structure also. So there are the one structure we call as trophoblast. This surrounding yang ni surrounding at here we call as trophoblast. Ini bukan trophoblast, ini we call as inner cell mass. Yang ini ada banyak cell at here we call as inner cell mass. Only the surrounding like this, only this surrounding all this we call as trophoblast. And inside the trophoblast, we also containing the fluid we own as blastocore. So actually this is a fluid lah, fluid. And then now once they already form the bastula or blastocyst acceptable bastula, so later it will undergo implantation, implant into the endometrium wall. So this endometrium wall, how is formed? This endometrium wall is formed during the menstrual cycle process due to that four hormone FSH, LH, progesterone, and also estrogen, especially progesterone and estrogen. Okay. So normally the inner cell mass, so the implantation is formed when the inner cell mass was starting to implant first. The facing is inner cell mass face to the endometrium wall, not the blastocore facing to the endometrium wall. Okay, the inner cell mass that locate at the one end will develop into the embryo proper, and then the bastula will reach the uterus, prepare for implantation. Now it's ready for implantation already. So you will see this lah. Okay, so this one is already implant. This one not ready, not yet, but this one the structure show is already starting to implant. Okay. Now we proceed to implantation. The implantation is occur when the trophoblast, the outer layer, the trophoblast of the blastocyst is initiated into the endometrium wall. Endometrium wall. And then the trophoblast will release the enzyme to break down the wall molecule of the endometrium wall, allow the invasion of the blastocyst. So if you want to make the one cell penetrate into the another cell, like awak kena macam penetrate lah, awak kena masuk dalam. So this is a wall, so you need to secrete the enzyme to break down the wall first, so that the blastocyst can penetrate into it. And then the trophoblast then will extend the finger-like projection like this. It will starting to extend the finger-like projection cells, cause the trophoblast tissue can capture the capillary of the endometrium. Because this is still the cell, you need to get the nutrient and also blood, get the nutrient from the mother. So it will starting to extend its cells until it reach the maternal blood vessel, so that it can receive the oxygen and then receive the nutrient, all that lah, to further development. Okay. At the same time, your inner cell mass will form a flat disc with the inner layer we call as inner layer, and inner layer we call as epiblast, and another layer outer layer we call as hypoblast. So your inner cell mass will further develop into the inner layer, inner part we call as epiblast, and then the outer layer we call as hypoblast. And then the trophoblast will continue expanding into endometrium wall and to form the four member we call as extra embryonic membrane. Extra embryonic membrane. Later we will see it. All these function, epiblast, hypoblast, and extra embryonic membrane have their own function. Later we will see it in during gastrulation process. So extra embryonic membrane will enclose the specialized structure that locate outside the embryo. Okay. So this is the process of implantation. So that's all. Once the blastocyst already fully penetrate into the endometrium wall, and then the trophoblast already extend itself until it reach the maternal cell wall, receive the blood, receive the nutrient from the mother, and then the inner cell may further develop into epiblast and hypoblast. Epiblast is inner layer and hypoblast is more upper layer. That's implantation is end. Okay. Next, we will go to the gastrulation process. Okay, so any question so far? No question? Okay, very good. Okay. After we go to the gastrulation, okay, gastrulation is actually the starting the development of the embryo already. Because it start process with the blastocyst become the 3-layer embryo, or we call as gastrula. So later we will form the 3 germs layer. So we will see how it form this 3 germ layer. So in human, the embryo is developed almost entirely from epiblast, meaning it's inner cell one. You see this diagram. This diagram is actually more complicated already lah. So see this epiblast is at here. This one is actually inner cell main structure, right? Inner cell main structure, and then this one is actually blastocore structure, and then this one, the outer one, is trophoblast. And then you see that the trophoblast now is expand until it reach the maternal blood vessel already, and then this one term, this sentence, I just want to tell you. I'll tell you that normally embryo is developed almost or entirely from epiblast. Hypoblast will not very play the role in embryonic development. So some of the epiblasts will remain as ectoderm, and while others will flow along the primitive streak to form the mesoderm and endoderm. So this is the 3 layer I say: ectoderm, endoderm, and also mesoderm. Okay, so this structure I cannot say that this is a further development. So actually in further development, actually amniotic cavity is starting forming, and then yolk sac is also starting forming, and then extra embryonic mesoderm cell for epiblast, and also chorion also starting forming. So this one I give you this. So normally at the end of gastrulation, 3 embryonic germ layer are formed. So the 3 embryonic germ layer I say is mesoderm, endoderm, and also ectoderm that I see here. These 3. So how to know that? Mesoderm is very easy at center. Middle one, M is middle, mesoderm. Mesoderm at the middle one. So you see this red color one is we call as mesoderm because it at the middle part. And then all of these three cells are developed from epiblast, the inner layer one. Epiblast, this epiblast. All 3. Hypoblast does not take over in any germ layer. You need to remember that only epiblast only. Okay. So ectoderm is the inner one. Endoderm is the outer one. So please remember ecto inner, meso middle, endo outer. Endo outer. Remember like that, endo outer. Endo outer. Okay. And then not only that, the hypoblast. Okay, so if you say that epiblast is already developed into these 3 layers, so how about hypoblast? Hypoblast will develop into yolk sac. This one, yolk sac, like this, yolk sac right here. Okay. And then epiblast not only develop into 3 germ layer, they are also developed into amnion. So amnion cavity lah at here, amnion, this one. Amnion play the role in secretion of amniotic cavity. Later you will it is considered as one type of the extra embryonic membrane too. And then extra mesoderm and 4 extraneous membrane now surround to the embryo. And then the trophoblast, especially trophoblast, will contribute to placenta. So you need to know the origin of placenta is coming from trophoblast. This is a keyword lah. Okay. Okay. So once the gastrulation, the end of the gastrulation is, it will form this 3 germ layer. Once these 3 germ layers are formed, then gastrulation is end, stop. Next, it will go to oogenesis, formation of the organ. But actually, this is very complicated process. So I just take from the Campbell. So in Campbell, normally it will develop from the about this from in the floor. Okay, but in the floor. Okay, but before that, this one is very important, especially in objective question. It will coming out every year. I think can say every year it coming out, because in human, oogenesis will be developed from these 3 germ layer: ectoderm, mesoderm, and also endoderm. Inner layer part, middle layer part, and also outer layer part. So process of 3 embryonic germ layer will develop into different organs. So I cannot say anything, you just memorize only. Ectoderm normally will be developed into nervous system, all the nervous system, nervous organ, and then also skin, and also your nail, and also pituitary gland are in the ectoderm. And then the endoderm normally is know about the digestion and also respiratory system. But all others are about mesoderm, like notochord, your skeleton, your muscle, your circulatory system, reproductive system, excretory system, inner layer of skin, all others are within the mesoderm. So you need to memorize it lah. Memang kena memorize. Because in objective question, that is very famous question that it will maybe highly possibility to ask one. Okay. Okay. Next is about the origin process or the beginning process of organogenesis. Because organogenesis is actually very complicated process. I cannot not in our syllabus actually, but in our syllabus we will mention about the origin of the organogenesis. So the organogenesis origin process is to formation of the notochord and also formation of the notochord and also formation of the neural tube. Okay. Okay, so normally the formation of the organogenesis, it will begin with the neural plate formation first. So it is a neurulation is an early step in the formation of the brain and the spinal cord. So firstly, though this one is the end step of your gastrula. So in gastrula you already contain ectoderm, mesoderm, and endoderm, and also containing notochord. So notochord is developed from the mesoderm cell. So you see this mesoderm cell is at the middle one. It will form the notochord. The dorsal, the belakang, at the belakang of the mesoderm cell, it will develop into the notochord. So the notochord is the rod that extends along the dorsal side of the embryo. And then the mesoderm cell will secrete the signaling molecule to the notochord, causing the ectoderm at the outer layer one, ectoderm become to neural plate like this. So it will formation of the neural plate is thus the example of induction, a process in which a group of cell or tissue influence the development of another group through the close range interaction. Okay, so actually neural plate will affect another type of development in the organ one. So neural plate is very important. Okay, because important. Okay. So once the neural plate is already formed, the cells in the neural plate will change its shape and curve inward. Curve inward like that. Curve inward. And then it curve inward and then roll itself into the neural tube. So now it will form starting to form the neural tube formation. So the second step of the organogenesis is to form neural tube. So the neural tube will run along the anterior posterior axis of the embryo so it will run inside and to the embryo to form the neural tube, and then the neural tube will become the brain in the head and the spinal cord along the base of the body. Okay, so vocal cord normally will disappear before birth, also although parts persist as the inner portion of the disc in the adult part. So that is why neural tube formation is very important, because it is the key organ that form the brain. So neural tube is come from where? From the neural plate. Neural plate come from where? Neural plate come from where? Come from ectoderm. Come from the ectoderm one. Ectoderm. Okay. Ectoderm. Okay. Okay, next, after that, once the neural tube is already formed, cell migration is the long range migration near the neural tube and including two sets of the cell. So first set of the cell is about the nucleus cell. There are another cell we call as nucleus cell. So normally this cell is developed along the border where the neural tube pin off the ectoderm. And then the nucleus cell will migrate to variety of tissue, including the peripheral nerve and also teeth and also bone. And then another set is we call as somite at here. So it will form the somite, which are the form where the mesoderm cell is lateral to the vocal cord, and then the somite will dissociate into the mesoderm cell, and then the some mesoderm cell will develop into vertebra. Okay, so in these terms that you need to know that after that there are the two cells we call as nucleus cell and also somite cell will develop into the different organ. So normally nucleus cell it will develop into the nerve tissue, teeth and also bone, and then the somite later will develop into the mesoderm too. So that's all for gastrulation process. Next, I will go through the placenta. Wait a minute. I think maybe I stop here now. Now it's already 9:20. So I stop here. Okay, I will proceed next week, next Tuesday, and finish up the reproduction in human topic lah. So I think that's all for today class. So any questions so far until for the lesson? Okay, if no questions, so I will end the class today. So we will see you next Tuesday. So thank you and goodbye. Have a good day. Bye.